This is a production of Cornell University.
Good afternoon, everyone. It's my pleasure to be able to speak with you and tell you the work that I have been doing. My work mainly focuses on plant virus movement and the acid transmission. As you know, the plant virus is very small, with a very small genome, and just encoding less than 10 proteins. However, the virus life cycle requires very complex activities, and the virus has involved a lot of complex mechanisms to ensure that all the activities can be performed. So today, I'm going to tell you about 3 genes from potato leaf virus that regulate a multi activities that are very crucial for the virus survival in plant host. The potato leaf virus is the poliovirus genus in the family of Lutividae. The natural host of the virus is potato, and it can also infect several other plants such as hairy nightshade, cicely, tobacco. The typical symptom after infection with this virus is leaf rolling, tuber necrosis, and also the chlorosis on the infected leaf. This virus is flown, preferred, and transmitted by acid in a persistent manner. Here I'm showing you a picture that we construct a virus-induced gene silencing vector using the leaf virus. And when we silence the TDS, we can see a lot of branch area. That area represents the localization of the virus. The virus is really flow limited. Here is the genome structure of leaf virus. It looks very simple, and it is a monopeptide linear signal strength RNA virus. Also, the whole genome can transcribe 2 subgenomic RNA named subgenomic RN1, subgenomic RN2. This simple genome use many translational strategies to encode its protein. For example, the read-through mechanism, frame shift, liquid scanning, internal ribosome intersite. So today, my talk will mainly focus on the read-through mechanism and the protein encoded from the subgenomic RNA1. Here's the subgenomic RNA1 encoded protein. There are several open reading frames along the subgenomic RNA1. For the OS3, the encoder code protein is the major capsid protein, and the whole wiring is composed with 180 code protein. And this protein has been reported to take part in viral transmission and movement. For the OS4, the encoder movement protein, this protein is related to virus movement, cell-to-cell movement, and systemic movement in the plant host. For the OS5, there is the RTD domain, named as read-through domain. This domain contains 2 parts. One is a very conserved N-terminus domain. Another is a very vulnerable domain with disordered sequence. This domain has been reported to take part in viral transmission, also take part in the virus movement. For the read-through protein, RTP is the read-through of the CP stop protein and generating a very large protein, about 75 kilodaltons. This protein has been very extensively studied. Our former work found that this protein functions in chains in flow mutation. This protein is on the surface of the structure. You can see this is the RTD domain. So here's the outline of my talk. In the part 1, I will introduce the translational read-through process. We discovered a downstream translation enhancer. The structure of the translational enhancer is critical for translation of RTP. And the translation enhancer and the cysteine-rich element, the interaction between these two domains is very important for read-through protein translation. In the part 2, I will introduce the read-through protein section. So here the proposed working model for read-through was stop coding. When translation happens, stop coding, the release factor will come and will arrive in the stop coding. Then the ribosome will get released, so the translation will get termination. However, when formation a very complex factor structure near the stop coding will inhibit the release factor coming, and then the residual stop coding will continue, and that means residual translation. And so a previous work 20 years ago, using a related badly yellow draft wire, they found that there are two domains named cysteine-rich region adjacent to CP stop coding and translational enhancer located within the RTD domain are very important for read-through translation. When they deleted these two domains, the RTP protein couldn't be detected. However, there is no further information about the mechanism and how the read-through are regulated. It's later unknown until now. So in our research, we will focus on studying the mechanism between these two regions. Firstly, to discover whether there is a translational enhancer in our leaf-roll virus, a serious mutation was constructed. We found that when we and the RTP expression was detected by Western blot using the agro-infiltrated benzimiana leaf. When we delete the nucleotide from the 4854, the RTP translation was severely impaired. However, when we deleted the nucleotide from 4933 or from the 5007, the translation of read-through protein is later affected. Also by inframe deletion, we found that the nucleotide between the 4854 to 4933 is the minimal region for the read-through translation, and these regions are considered as the translational enhancer of leaf-roll virus. So to ask the translational enhancer section is determined by the nucleotide in the translational enhancer or the amino acid encoded from this region. So a stop codon was introduced before the translational enhancer, and making the nucleotide after this will not be translated. The RTP was detected. We found a truncated RTP, indicating that the nucleotide in this region is determined the read-through translation, not the amino acid translated by the translational enhancer domain. Also, a dark RTP mutant, which all the nucleotides after the CP stop coding was deleted. As this mutant was used as a negative control. As shown in this figure, we couldn't find any RTP that read-through translation. So next, the nucleotide alignment of homologous translation enhancer sequence among several polyovirus was carried out, we found that the nucleotide similarity is very low and it's less than 40%. If we looked at the secondary structure of this region, we found that the secondary structure is very similar. And there are several double loops in this region. So previous work on other systems found that the double loop structure is very important for read-through or stop coding. So to study the double loop structure section in our read-through translation, we focused on studying 3 double loops: double loop 1, double loop 2, and double loop 3, and we studied these double loops separately. For double loop 1, we introduced 2 mutants, which the stem will be opened up, and the RTP translation was detected by Weston. We found that the translation was severely impaired. However, when we made a complement of the sequence here and made the stem close, and we found that the RTP translation will get reversed translation. Also, we introduced another mutant, which the loop of the double loop 1 was closed, and the RTP translation was also impaired. So this dot here suggests that the double loop struct 1 is necessary for RTP translation. We also studied the double loop 2 and double loop 3 on the sections of read-through translation, and we found that there's no effect on RTP translation after disrupting the double loop 2 and double loop 3. Next, we studied on the characters required for the read-through translation required for the double loop 1 in read-through translation. Firstly, we focused on the plasticity, and we changed the double loop size by 1 nucleotide or 2 nucleotides. And the read-through translation can be detected by Western blot, indicating the size of the double loop doesn't matter for the read-through translation. Also, next we studied the position and the sequence between the stop coding and read-through translational enhancer. Firstly, we deleted the nucleotide between the stop coding and the translational enhancer, and generating a 500 nucleotide decreased RTD mutant. We could find the RTP translation can be detected. Also, when we replaced the nucleotide between these 2 regions with the foreign HA tag sequence, although the RTP can be detected, indicating the position and sequence between these 2 regions is not so strictly required for the read-through translation. So I have told you that formal work found that the stage element and the translational enhancer is very important for related virus read-through translation, and we found that there is a very similar secondary structure between the 2 related virus. To check whether the stage regions of our retrovirus also required for read-through translation, and to see whether or not there is a communication between the stage element and the translational enhancer, and whether this interaction is virus-specific. A several chimeric mutant was constructed as shown in this figure. This is what type of retrovirus. We could detect the RTP translation. However, when we deleted the stage element, of the leaf roll virus, no RTP could be detected. When we replaced the leaf roll virus cleavage region with the related CYD cleavage element, there is also no RTP can be detected. Also, when we replace the leaf roll virus translational enhancer with the related CYD translational enhancer, no RTP can be found. However, when we replaced both cleavage region and the translational enhancer with the CYD related sequence, we can find RTP can be translated, indicating that communication between translational enhancer with the cleavage domain is virus-specific and indispensable for RTP translation. To see, next we asked what the effect of RTP mutant on infection of the plant. So the leaf roll virus titer was measured in systemically infected leaves, and we can find all the mutant can get systemic infection as early as 3 weeks. However, with the RTP translation, all the viral titer increased by time. However, for the without RTP expression, the viral titer decreased from 3 weeks post-inoculation. And for example, the delta RTP mutant, you can see the titer is always decreased to a very low level. However, we found there are viral titer switch using the loop1 mutant. The titer of virus switch to increase in 6 or 7 weeks post-inoculation with this loop1 mutant. So to see what happened on this mutant, whether or not this sequence are still kept integrated as constructed. So RT-PCR was carried out to check the sequence integrated of this mutant. Firstly, we detect the mutant sequence from 3 weeks post-inoculation leaves, from 3 weeks post-inoculation leaves, and we found that all the mutants still keep integrated as constructed. Here is the wild type secondary structure about the translational enhancer, and you can see all the mutants secondary structure of the loop 1 was still being impaired. However, when we detect the sequence from 8 weeks post-inoculation infected plant, we found they all have additional mutation. And we found that a compensatory mutation that would restore a pseudo W1 structure was found. Although the W structure is a little different with the wild type, as shown in this figure, this restores W1 structure. So to see whether this sequence is working, you will restore the residual translation. So we made this mutation into the retrovirus infection clone and infiltrated into benzimiana leaf and then detect the RTP translation. We found that all the mutants can restore RTP translation, indicating that the W structure is very important for residual translation. And also, the structure is in a very strong selective pressure to maintain the RTP translation. Here is the conclusion of part 1. A translational enhancer located 640 nucleotides downstream of the CP stop coding is required for residual translation. W1 of the translational enhancer is required for efficient RTP translation. But there is plasticity to its structure, communication between the translational enhancer and cysteine-rich element is required, and virus-specific, but the length and makeup of the sequence between them is not important for read-through translation. There is a strong selection for mutation that restores a subgroup 1 structure that allows RTP translation. Now that we found that the RTP can be recovered translated, so it might have very important function in the virus survival. So in the part 2, I will introduce our study on the read-through protein function, and we found there is an interaction between the different virus and P with the read-through protein. And finally, a model was rigged to explain the RTP and P interaction. So all the phloem-limited virus, after they get inoculation into the phloem tissues by aphid or some other insect, they have to overcome 2 barriers to make sure they can establish the successful infection, the successful infection of the entire plant. Firstly, they have to get replication in the inoculation field. Second, they have to move out from the replication site, both in the cell-to-cell level, and also they have to move in a long distance level. And they have to move to the phloem and get systemic infection, and then exit from the phloem and start new infection. To see whether the RTD domain has an effect on different virus replication, a serious RTD domain was constructed, which this mutant, the RTD protein, was seriously impaired. As shown in this figure, we found that the RTD domain had no effect on virus replication, both in 3-day post-inoculation and 5-day post-inoculation. This result excludes the possibility that RTD domain had the effect on virus replication. As I showed you before, we can detect the systemic infection of the RTP mutant as early as 3 weeks, indicating that absence of RTP doesn't prevent systemic movement of the virus through phloem. So next we want to ask whether RTP has the effect on the cell-to-cell movement of this virus. So tissue print was performed to detect the cell-to-cell movement in the phloem tissue. Here, with the RTP expression, we can see a lot of infection foci in the phloem tissue, both in this is the stem, this is the cross section of the leaf. We can see a lot of infection foci. However, without RTP expression, the virus is present, but the infection foci is very limited, indicating that the RTP protein had some role in the cell-to-cell movement of the leaf-4 virus. So the MP protein is the movement protein of this virus, and our framework found that the number of infection foci are also decreased in tissue infected with the leaf-4 virus mutant that doesn't translate MP. The similar phenotype between the RTP mutant and the MP mutant inspire us to study whether there is a functional interaction between the RTP and the movement protein. So first we studied the localization of these 2 proteins. For MP is the movement protein. It can localize to the plasmodesmata, and the 30S promoter agro-infiltrated benzimiana leaf. You can see the PD localization of MP. When we express the RTP and the 30S promoter we found that RTP can localize to the cytoplasm and also in the nuclear layers. However, when we express RTP with the infection of the virus, the localization of the RTP changed. It switched to the plasmodesmata. Obviously, there are some other proteins encoded from the virus changed the localization of the RTP protein. So because MP has the intronic property to localize to the plasmodesmata, so next we detect the localization of RTP without MP translation. We found that RTP doesn't localize to plasmodesmata when in fact with MP deficient mutant, indicating the MP protein is a protein which helps the plasmodesmata, helps the RTP protein to the plasmodesmata. Next, we want to know what the mechanism of this process, how the MP makes the RTP to the plasmodesmata, whether there is a direct interaction with the two proteins, and then the MP drags the RTP to the plasmodesmata. So we also detect MP alone. When we express MP alone can direct the RTP to the plasmodesmata, indicating only MP alone can help RTP to the plasmodesmata. Therefore, we studied the interaction between MP and RTP to support whether MP drags RTP to plasmodesmata. So the BIFC assay was carried out to detect the RTP and the MP interaction. If these two proteins can interact with each other, we can see the yellow fluorescent and confocal microscope. Here is the BIFC result. We confirm the interaction of RTP and MP, and we use the CP, virus CP protein as a positive control. So to support our idea that MP drags RTP to the plasmodesmata through binding, by mutagenic analysis, the region of RTP responsible for the interaction with MP was found out, and we found that there is no interaction between this mutant with the movement protein. This is the positive control wild type RTP. So next, we want to know what the localization meant. We found that the mutation that disrupts RTP-MP binding prevents the localization of RTP to the plasmodesmata, both co-infected with the virus infection or the MP alone. This result supports the idea that MP helps direct the RTP to the plasmodesmata through binding with this protein. So next we construct this RTP mutant into the virus infection clone and inoculated into the plant. We found that we detect the virus after 4 weeks post-inoculation. We found that fewer infected loci observed in tissues systemically infected with this RTP mutant, and this phenotype is very similar with the WF1 mutant, which the RTP couldn't be translated, and corresponding with the limited infection loci, the titer of the virus is also very low in the systemic leaf. This result supports that RTP helps the virus cell-to-cell movement depending on the binding with the movement protein. So here is the conclusion of part 2. The RTD domain of the virus has no effect on virus replication. RTP is not required for long-distance movement, but it facilitates systemic spread. RTP interacts directly with MP, binding to the MP redirects the RTP to the plasmodesmata. RTP appears to function to move before virus between flown companion cells. With still unknown whether the cell-to-cell movement requires structural RTP or soluble RTP. Here is the whole summary of my talk. After the leaf-4 virus gets inoculated into the plant cell, firstly they have to translate the replication-associated protein and make the leaf-4 virus get replication. Also, they will incur some proteins such as CP and MP. For the RTP, for the read-through translation, there is a translational enhancer downstream of the stop coding, and it can form a double structure, and through communication with the cysteine-rich domain, inhibit the release factor coming, and then the translation will continue, the read-through protein will get translated. After this protein gets translated, it will be recognized by the movement protein, and at the help of the movement protein to reach the plasmodesmata and help the virus move to the neighbor cells. We still don't know whether MP move to the plasmodesmata associated with the virus. Also, there are some other possibilities that the MP just interacts with the soluble RTP, and they move to the plasmodesmata, and then modify the size of the plasmodesmata. When the virus come to the plasmodesmata, this modification will facilitate the virus move to the neighbor cells. So as we can see, the virus is very simple, but it works in a very sophisticated manner. They work together both in the nucleotide level and in the protein interaction level. So here is the acknowledgement. I thank all the members from Student Grade Lab. Thank you so much for your help in my project and helped me improve the talk. And also I thank the collaborators from Michelle Stillier's lab. Thank you so much for the discussion. I also thank all the guys from the greenhouse. Especially thanks to Raj for helping me prepare all the plants. Finally, I thank the funding. Without funding, we can't do anything. Thank you for your attention.
This has been a production of Cornell University, on the web at cornell.edu.